Hello, RDs and RDs to be. Welcome to the RD Exam Ready podcast. My name is Zach Caseberg, your tutor, coach, and guide along the path to passing the RD exam. And my biggest passion is helping you, RDs and RDs to be, to learn more about this exam so you can dominate this thing. You've likely heard in previous episodes with similar topics, but one thing that always fascinates me is the story of the world that exists at your cellular level. It's truly what differentiates a dietitian from a nutrition, a life coach, or whatever, is that we're quite keen on knowing the physiology of things. And in a world of plenty of voices, ask anyone, and they're probably going to tell you that protein is important. But how is protein important? Protein and amino acids themselves, how do they get to themselves? What happens if I get too much and what too little? We'll discuss all of that even more as we dive into the biochemistry of protein, our major macronutrient. Now, we've got to warm up with the basics of protein here first. We know protein is essential to so many functions of the body. The creation of enzymes, cofactors, hormones, transporters are all made of protein. Protein is a buffer in your body. It can act to balance fluid, it can act to balance pH. Our immunoproteins and immunoglobulins like IgE and food allergies is made of proteins, and proteins has so many number of functions. Now, the protein structure, we want you just to be a little familiar with this for exam day, but don't need to know the exact kind of function or structure of every amino acids. Every amino acid, it has a central carbon. And off of that, it's going to have at least one amine group, and that will be that dash NH2 or NH3 group. And then it's also going to have one carboxyl acid group, COOH, that you're going to see on there too. Now, within that COO and that NH3, they're very nice because they allow this amino acid to take hydrogen ions, to give hydrogen ions, and it's a lot of what can make a different amino acid a different function of itself. So, this is just a good thing to kind of know that they're all going to have this basic backbone, but that R chain or that R group can really differentiate the specific amino acid itself. Amino acids can have different polarities too, protein molecules can. And the polarity is largely just dependent on that side chain or the R group. And we have both nonpolar and polar amino acids. Much of these structural properties, like polarity, is what helps to categorize many of these amino acids as being either essential, meaning we have to get them from the diet, or non essential, meaning we can synthesize them or make them in the body. Essential versus non essential amino acids. If I asked you to give me a list of your essential and non essential, how do you think you would do? Well, it's a lot of pressure. No big deal. Let's do it together, right? And let's talk through what the essential amino acids are. There are 9 of them I need you to be familiar with. We have phenylalanine, valine, threonine, tryptophan, methionine, histidine, isoleucine, leucine, and lysine. These are our 9 essential amino acids. Now, there are several mnemonics that exist out there. I always say, create your own if it helps you to kind of go through these. I do want to caution one thing, and maybe I'm thinking way too much in the logistics, but there's always been this private Tim Hall that's kind of been this mnemonic that's used all the time. If you look that up, even an image of it, one thing you might see is that private Tim Hall, the mnemonic itself, has 10 letters in it. So that makes me confused because there's 9 essential amino acids. So I've kind of adapted this one a little bit myself, and I just want to be clear with all of you. So the mnemonic that I would maybe use is use that PVT, the private, there's our first 3, TM, or kind of like Tim, just without the I, and then use the Hall after. That's going to get you 9 amino acids here. So PVT, TM, Hall. Okay, that could be kind of what we use. Or let me back that up. Okay, so PVT, TM, Hill. Okay, so PVT, TM, HILL. That's going to get you all of your essential amino acids of phenylalanine, valine, threonine. There's your PVT. Tryptophan, methionine, there's your TM, and then histidine, isoleucine, leucine, and lysine. Those will get you your hill at the end there. So if that's a helpful mnemonic for you, definitely use that or come up with one that really makes a good fit for you if you can find one there. Now, with the non essential amino acids, we need to know there's 11 of these, okay? 11 amino acids that are non essential that you're going to have to know about too. Your non essentials are going to be tyrosine, cysteine, asparagine, aspartic acid, glutamine, glutamic acid, glycine, arginine, proline, aline, and serine. So 11 of those to know. For a mnemonic, let's group together similar letters. I think that'll be easiest for me to put the A's, the G's, the P's, S's all together, right? Here's my mnemonic I came up with. Angry apes always ate gentle green grapes prior to cold soup. Okay. So that can be a little mnemonic for you to go through those 11. So angry apes always ate gentle green grapes prior to cold soup. See, you can come up with all the mnemonics you want. You can, use different AI functions to help you come up with some. Be fun and come up with things that are relevant to you and your likes. Now, I do need to mention there is a third category known as conditionally essential amino acids. I'm not going to make you remember these here, but let's go through an example so you just know what we're talking about. Conditional is exactly how it sounds. There are going to be cases in which the body cannot synthesize an amino acid that is normally non essential, and thus that non essential amino acid now becomes essential, right? PKU, or phenylketonuria, this is my favorite example of this. It's an inborn error of metabolism, usually affecting infants, very, very young children, and it's caused by a dysfunction in the enzyme that takes your essential amino acid, phenylalanine, and converts it into tyrosine, a non essential amino acid. What happens when that enzyme is dysfunctional? We can't make the tyrosine out of anything. So we need to get it from the diet. It now becomes conditionally essential because we couldn't make it anymore. In previous episodes, we've run through all the basic enzymes involved in the digestion of all of our nutrients. Let's do a quick rundown of digestion of protein and really focus in. Protein will be ingested and first start its digestive journey in the stomach, right? This begins with hydrochloric acid found in the gastric juice. It starts to denature that protein structure so that quaternary, tertiary, and secondary structures of the protein are being broken down by hydrochloric acid. But we still have peptide bonds that are not broken during this process. Pepsinogen, an inactive enzyme, becomes its active form of pepsin. And what happens is the pepsin then breaks peptide bonds, leaving us with a bunch of polypeptides or chained together proteins. But now they're not so bound together so tightly. Once that acidic chyme mixture moves into the duodenum, we know that two hormones are going to be responsible for helping here. Secretin, the hormone that's responsible to tell the pancreas that the pancreatic bicarb needs to be released, and CCK, or cholecystokinin, the hormone that tells the pancreas to digest and release enzymes into the small intestine. Just related to protein, though, the pancreas is releasing several zymogens, as they are known: trypsin, chymotrypsin, just a few examples. I don't think you're going to need to know all the different types of zymogens on the exam, but just know kind of what a zymogen is, that it cleaves, it breaks peptide bonds, and helps release these free fatty acids, or excuse me, free amino acids in short peptides that can then be absorbed into the GI tract. We've got to dive into the biochem. That's our big goal today. Your amino acids have now been absorbed after you've eaten that meal, and they've entered the portal blood to be transported to tissues. As we talk about tissues, I do want to talk about amino acids and just a bit how they're being used in the body. One important difference between protein and other macronutrients is that protein is not readily stored in the body for energy production, right? A lot of the muscle, your muscle on your body, it's a storage form of protein, but not for energy production. We're not readily breaking that down like we do glycogen or body fat or something like that, right? So, amino acids can be used to make energy, and this gives protein a very special role during states of fasting or starvation, for instance. The first organs in which the intestines are gonna and excuse me, the first kind of area where the amino acids are really gonna pass through is primarily gonna be the liver, because the liver will be central in controlling and monitoring the amino acids to adjust metabolism and use. Now, your muscles in particular, they like protein, but they really love branched chain amino acids, that valine, leucine, isoleucine. The muscle can actually readily use these BCAAs for energy because they have a specific enzyme that allows them to readily use those BCAAs to be oxidized and provide energy. When protein is breaking down within the body, when we're metabolizing it in order to get rid of its waste, or we're going to try to get some energy out of it, that's really what our goal is. Remember that catabolism is breakdown. When we're talking about breakdown of the proteins, a lot of the times we're trying to process them to either make energy out of them, of course, or two, we're trying to process them into something else. Deamination is the removal of an amino acid group from the amino acid. There is no transfer going on during deamination. What you're doing is you're removing that kind of group, and what you're left with is known as this carbon skeleton, also known as a keto acid. That's what deamination is just when you remove that group. Transamination is when you're going to deaminate amino acid, but you're going to transfer that amino group from one amino acid to a keto acid, okay? Transamination is the process of where you're making new amino acids. In particular, your non essential amino acids. This is how you're doing that. After transamination, you might have that carbon skeleton left over that used to have that R group. You might also have some NH3, some NH4, some ammonium that is coming and breaking off from this transamination process too. That carbon skeleton is going to be used for a few things. Some of it can be gluconeogenic, meaning we can take this leftover carbon skeleton and we can convert it into glucose throughout the body. Acetyl CoA can be one thing that can be made as well from this, and we can make that into the TCA cycle, right? So take that kind of backbone, that carbon skeleton, and then take that, make it into acetyl CoA, and then run it through the citric acid cycle. Other substrates could be also made for the TCA cycle out of this, so it's not just acetyl CoA. Couple other things you could do. You could make this carbon skeleton into ketone bodies, perhaps, and you can kind of use ketone bodies for energy if we were in a state of starvation. We could also convert these carbon skeletons to cholesterol. Um, and one more point that we always remind people is you can overdo the protein, right? So if you have excessive amounts of protein in the diet and you have filled up all of your needs for everything, well, your protein is going to be converted to fatty acids, and then it's going to be stored as adipose tissue. So that's just a really good point to remember that you can overdo it with the protein. Key point here, RD, transamination requires vitamin B6 for much of this process. So be very familiar that B6 is going to be essential. Key point for exam day. Another area I need you to understand about catabolism is that we have these byproducts that come from things that we need to also know how do we get rid of them. Ammonium is that NH4 that breaks off from this deamination process, right? So when we go through deamination, you're left with this ammonium. And this ammonium is not good for the body. It's really not good to have a ton of ammonium in your blood. So you want to get rid of it. And there's a few ways that we do that. We're going to introduce the urea cycle here so you know what we use that ammonium for. The urea cycle is how we remove that excess ammonium from the body, and it takes place in the liver. What happens is the ammonium will enter the urea cycle, and much like Krebs, it goes around this circular cycle, and it left over we have urea. Urea is your end product. What's nice about urea is less toxic, and it can then travel in the blood to the kidneys to be excreted in the urine. We know normal levels of BUN in the body of our kind of blood urea nitrogen is going to be about 8 to 20 milligrams per deciliters. So now we just need you to connect the dots of we know the liver is involved with the urea cycle. Okay, what happens if the liver has a disease? We know what the kidneys do to get rid of urea. What happens if we have kidney disease? So think about that as you're going through your clinical sets. Anabolism is the other side of protein, kind of that buildup, right? Building blocks of enzymes, muscles, and different tissues through the body. We do require ATP to do this process. We don't need to know a ton about anabolism in this case, but anabolism of protein is really going down to anything we're making. And what it requires is transcription and translation. Remember those back from biology? We don't need to know the exact formation and all the things of transcription and translation, but let's understand it a little bit. We're taking those amino acids that we've all talked about here, and in transcription, we're kind of assembling a strand protein, right? We're assembling these things to make this mRNA strand, this long protein strand. And over time, what you're going to do is you're going to take those chains of amino acids and form them into this kind of sequence in the nucleus, this pattern, right? And this is so that I can make different byproducts. This is so I can make proteins and muscles and all the things in my body is I'm using these as the building blocks for these amino acids. Translation is what happens second. That's when that formed RNA is stranded together to make it into a formed protein. And that's how you're making the protein in the protein tissues. Now that you've gone through the biochem, I just want to give you more tips as far as studying it. We've focused a lot on the basics, and it may not feel like basics when you're studying it, but trust me, the exam only needs us to know the major key points. That's good news. Remember for biochem, what you need to know is what are the inputs, what is happening in between, and what are the outputs. We don't need to know every step, inputs and outputs of the key process. We need to know where it happens in the body, we need to know what triggers it, and we need to use tools to help us out. Draw it out, use note cards, use mnemonics, create visual flow charts in all types of ways to help yourself. Record yourself talking, and hey, tell a story to really go through this process of knowing this. Now let's apply these to 3 practice questions. Which of the following is not an essential amino acid? A, glycine, B, threonine, C, phenylalanine, or D, lysine. Which of the following is not an essential amino acid? Glycine, threonine, phenylalanine, or lysine. Okay, so I'm going to use my PO, my process of elimination, to help me see which ones am I confident about, which ones am I questionable on, which ones do I really don't like. So which one is a not? Okay, not being the key point. Not an essential amino acid, meaning we're looking for a non-essential amino acid here. Starting with A, glycine. Well, I go through my PVT, TM, Hill in this case. I don't see a G in there. So to me, glycine seems like it's probably a good answer. It's probably going to be a non-essential amino acid. I'll put a little check mark by that one now. For B, threonine, well, that's going to be an essential amino acid. I know it's one of my 9. Same goes for C, phenylalanine. I know that fits into my private Tim Hill kind of a situation. And then lysine. Lysine is also one of my essential amino acids. So I would say B, C, and D are all essential, and I'm going to get rid of those. That's leaving me just with answer A as glycine as being the one that is the following that's not an essential amino acid. Number 2. The liver has the ability to convert blank to blank through the urea cycle for later excretion. A. Ammonia acetyl CoA. B. Amino acids ammonia. C. Urea ammonia. Or D. Ammonia urea. Let's read through each and plug those in and see how we like for each answer. The liver, let's start with A. The liver has the ability to convert ammonia to acetyl CoA through the urea cycle for later excretion. I don't know about you folks, but I don't remember acetyl CoA being a big piece of the urea cycle here. I don't think this would be a good answer. We just don't really make acetyl CoA out of this, so it's just not a good fit, right? B, the liver has the ability to convert amino acids to ammonia through the urea cycle for later excretion. Hmm, maybe, but really what we're doing is I don't think we're converting the amino acids to ammonia in the urea cycle. I don't think that's what we convert them to. The amino acids aren't being converted, it's just that ammonia is a byproduct or broken off of an amino acid. So I would say A and B are both bad answers, and I'm going to get rid of those. C, the liver has the ability to convert urea to ammonia through the urea cycle for later excretion. Okay. Or D, the liver has the ability to convert ammonia to urea through the urea cycle for later excretion. I feel like answer D is my best answer here. The urea cycle will be when we convert our leftover ammonia to urea, and then it can then be excreted. I would go with answer D as my best answer for number 2. Number 3. During the process of protein catabolism, transamination results in A. Removal of the amino groups from amino acids without transfer. B. Conversion of carbon skeletons into ketone bodies. C. Transfer of amino groups to keto acids, allowing for synthesis of non essential amino acids. Or D. Elimination of excess ammonia in the kidneys. Okay. So during the process of protein catabolism, transamination results in which of these? Let's start with answer A. Removal of the amino groups from amino acids without transfer. I would say A is a bad answer first here because what we're seeing is in transamination, we're removing that group, but then we're attaching it to another thing, right? We're attaching it to another carbon skeleton keto acid to make another amino acid, right? So I know A is not a good answer for transamination. B, conversion of carbon skeletons into only ketone bodies. I'm going to put just a question mark by B for right now. I don't feel good or bad with it at the moment, but I want to check all my answers. C, transfer of amino groups to keto acids, allowing for synthesis of non essential amino acids. I think C is a great answer. This does describe transamination in the process by which we go through making those non essential amino acids. Or D, elimination of excess ammonia in the kidneys. I don't think it's D in this case. It doesn't sound right. So if I was between B, conversion of carbon skeletons into only ketone bodies, or C, transfer of amino groups to keto acids, allowing for synthesis of non essential amino acids, I would say C will be my best answer for number 3. All right, RDs and RDs to be, that wraps up the show today. I hope you listened, learned, enjoyed, and really just took something in from today's podcast. Remember, passing this exam is a journey. We all need assistance along the way. Continue to help yourself, help me, spread the word to other students that you know are studying that could use some of this assistance. Rate, review, help spread the word if you don't mind. And remember, as always, happy studying, RD to be.